Today, we will share the article titled Endometrial Stromal Cell Senescence, a Non-Negligible Factor in Recurrent Pregnancy Loss. Pregnancy depends on the dynamic remodeling of the endometrium, which requires satisfactory receptivity and balanced immune inflammation. Endometrial stromal cells, EnSCs, the core functional cells of the endometrium, complete proliferation, differentiation and decidualization during the menstrual cycle. The dynamic balance between their physiological transaging and normal differentiation is critical for early pregnancy, while uncontrolled aging impairs endometrial function. Based on the endometrial microenvironment, this review summarizes the physiological and pathological changes of EnSCs, and focuses on the molecular mechanisms of EnSCs aging and its interaction with decidualization, providing a new theoretic basis for understanding adverse pregnancy outcomes such as recurrent pregnancy loss. Highlight. From a temporal perspective, this article illustrates the dynamic pattern of EnSCs aging and decidualization, as well as their vital roles in pregnancy progression. It summarizes that multiple molecular mechanisms collectively mediate the pathological aging transition of EnSCs, and confirms that abnormal EnSCs aging is closely associated with various pregnancy outcomes. Furthermore, targeted interventions against cellular aging are expected to serve as promising strategies for the prevention and treatment of recurrent pregnancy loss. The platform provides 3 workflows tailored for analyzing metagenomic data: the reference-based workflow, the de novo workflow, and the binning workflow. It's worth noting that we utilize 12 widely used bioinformatics databases in metagenomic analyses to predict functions accurately. We present the primary software tools and visualization techniques used in assembly, binning, taxonomic analysis, and functional analysis, ensuring its suitability for a wide range of purposes and scenarios. External stress and mitochondrial damage promote reactive oxygen species, ROS, accumulation and DNA damage. They inhibit the cell cycle and induce cellular senescence through the ERK/AKTmTOR-FOXO1/P16 and ATMP53P21 signaling axes. Reduced NAD levels weaken sirtuin-1 activity. The TLRIK pathway activates NF-kappa B to upregulate the expression of SASP factors, including IL-1A, IL-6, CXCL1 and IL-8. Altered epigenetic modifications further stabilize the cellular senescent phenotype. Next, this article elaborates on the mechanisms and signaling pathways underlying endometrial stromal cell senescence, upper left, hormone pathway. PRB and PGRMC1 regulate FOXO1 to promote the secretion of inflammatory factors and SASP, thereby inducing cellular senescence, upper right, PGE2 pathway. Upstream signals boost PGE2 synthesis and activate the pKa pathway via receptors, upregulating senescence markers, lower left, oxidative stress. ROS activates multiple signaling pathways to trigger inflammation and cell cycle arrest, mediating cell senescence, lower right, metabolic/SIRT axis. Sirtuins combined with ATP inhibit senescence, reduce NAD and ADP suppress sirtuins activity and aggravate cellular senescence. Finally, this article further analyzes the interaction model between EnSCs aging and decidualization under physiological and pathological conditions. Under physiological conditions, endometrial stromal cells undergo normal decidualization. A small number of senescent cells and transient SASP can be cleared by UNC cells, maintaining endometrial receptivity to support embryo implantation. In pathological conditions, excessive EnSCs aging and impaired cell clearance lead to senescent cell accumulation and sustained SASP activation, which disrupt decidualization and ultimately cause implantation failure and recurrent pregnancy loss. Based on this mechanism, targeted regulation of EnSCs aging can repair residual damage, alleviate inflammation, restore endometrial receptivity and remodel maternal-fetal homeostasis, providing new targets and therapeutic strategies for the prevention and treatment of adverse pregnancy outcomes. Summary. This article focuses on the aging of endometrial stromal cells, EnSCs, and clarifies its vital role in regulating endometrial receptivity and maternal-fetal homeostasis. Multiple mechanisms jointly mediate the pathological aging of EnSCs, thereby impairing decidualization, triggering inflammation, and resulting in recurrent pregnancy loss and other adverse pregnancy outcomes. Targeted intervention against cellular aging and endometrial microenvironment exhibits great application potential, offering novel insights for the prevention and treatment of related diseases. Nevertheless, further basic and clinical studies are still required to verify its clinical translation value and safety.
I hope this brief overview will help you follow the PPT more easily and grasp the main storyline and clinical implications of this review. Thank you for your attention.